Hello, my name is John Titus, and I've had the pleasure this summer of being the mentor for the 4 Odum interns who've been working hard all summer on their project. And for me, this has really been a great experience. It's been a wonderful pleasure to work with these 4 really remarkable young people. And so I've learned a lot. Been a great experience. And when they first came, of course, they didn't know each other, and they're all really different from each other. I see the nods over there. And but they really worked together excellently. They helped each other work through their problems. They really bonded and became a unit. So that was really a wonderful thing to see also. So they really did fantastic work. It's really been, a blessing for me to be able to work with them. So anyway, so the first speaker will be Angela, and she's from New Jersey. She attends Cornell University. And are you a biology major?
Yeah.
Yeah. And she's going to have a beautiful presentation.
When the viewer flower Oh, okay. So, first, about me. I like hiking. This is me with my brother up in the Adirondacks. I like baking. I like painting. I like sunsets. And I like plants. And because I like plants, I also like pollinators. So this summer I studied native bees here at the hike. And before I dive into the specifics of the bees I found here, I just wanted to give a general overview of the types of native bees in general, in New York State. And there are basically 2 main types, solitary bees and bumblebees. So solitary bees are basically exactly what they sound like. They're bees that do everything themselves. So the female bee makes the nest, collects all the resources, and raises the young all on her own. Whereas bumblebees are colonial bees, so they form like little hives, around 80 bumblebees to a hive, give or take. And so they have division of labor. There's a queen, there are worker bees. And so this right here is a solitary bee nest. So they just kind of, some of them live underground and some of them live in like the hollow stems of different plants, like sumac or something. And then this is a bumblebee hive right here. So you see these little cups, I guess. That's where the young came out of, and it's also where they store their pollen. A non-native bee that everyone here is probably familiar with is the honeybee. And honeybees are also colonial bees like bumblebees, but they have huge hives of hundreds and hundreds. And so you'll see later on in the presentation that honeybees really affected my study just because of the sheer number of them, because each hive just has so many different individuals. And that's a cute little honeybee right here. Okay, so these are different native bees that I found at the hike this summer. So do any of you guys know what some of these bees might be? Yeah?
A green bee?
Yeah, a green bee! This bee is from the Algoclorini family, so all the bees in this family are green, and there's quite a few different types of green bees here at the hike. This one is Algocoropsis metallica, and you can tell because its legs are green. Here, all the other green bees have black legs. This right here is a small carpenter bee. It's from the Seratina genus. I don't know if you can see from the photo too clearly, but it's kind of a bluish green color. It's very small. These bees here are Hylaeus bees. They're the yellow masked bees. So everything in the Hylaeus genus is, has a little yellow thing on its face. This is a Lucia glossum bee, commonly known as sweat bees. Green bees are also commonly known as sweat bees. This is a megachilid bee. It's a leafcutter bee. This is a halictid bee, which I don't know, I have no fun fact for that. It's just a type of bee. And this is the tricolored bumblebee. So all of these are bees, and besides the bumblebee, all of these are solitary bees found here at the Huyck. It worked. So specifically, I wanted my research question to be about native and non-native plants. So native plants have a very long evolutionary history here, and so they've been able to co-evolve with the native bees in the area. I wanted to know if this long history makes it so that it's very hard for non-native, like European, Asian plants, to come and take the place of a native plant in the ecosystem, where it just won't provide services that the bees are used to and are able to get to. Okay, so to do that, I looked at 12 different species of flowers over the course of the summer. So on any given week, I had 5 or 6 different species of flowers that I was looking at. And then as the flowers bloomed and faded over the course of the summer, I changed, I dropped and added them to keep up with what the bees were able to access. So for each flower, I had 3 different sites. And at any given site, I would spend 15 minutes a day just like swiping and trying to catch all the bees I saw. So actually, I did not catch any honeybees or bumblebees because these bees I can just like look at and identify pretty easily. So I just recorded the numbers of those that I saw. But all the other bees, I would catch them. I put them in little tubes like this, and I bring them back here to the lab and look at them under the microscope and identify them to see which bees are going to which types of plants. So in order to answer my question about whether bees visit native flowers more than non-native flowers, I measured abundance, richness, and diversity. So abundance is just the total number of individuals that I found at a site. Richness is the total number of species I found at a site. So each species was counted as one. And diversity is richness plus a measure of species evenness, which is a measure of the distribution of the bees. So, for example, at many sites, as you'll see later, I had a lot of honeybees and like a few of the other bees. So that's not very good diversity because it's a lot of one thing and then not a lot of the others. And then I used the Friedman tests to test the significance of my results, and then I used the Spearman rank test to look at correlations. So this is the data that I got for the abundance. So these 2 graphs right here, they're the exact same graph. It's just that I grouped one graph by I color blocked one graph by the type of flower and one graph by the native and non-native status of the flower. So here you can see purple is native, yellow is non-native, and you can see very clearly right off the bat that the native flowers had much more visitation than the non-native flowers. So everything except for the exception of this one site, all the high peaks are native, and so it was very, very significantly so that the native plants had higher visitation rates than the non-native plants. This one site right here, it's the bird's feet, or Lotus corniculatus. I'm not sure how you guys know it here, but this site right here was actually just right out on the lawn, and there was a lot of honeybee visitation. Actually, all of these high peaks had a lot of honeybee visitation, and that's why their numbers are so high. And so it's just something to consider that native plants are attracting not only non-native bees, but also the non-native honeybee quite heavily. So this is the richness. You can see that, so this is the species counts. So even when you're not looking at individuals, the native plants are attracting higher amounts of species as compared to the non-native as well. So there are more different species of bees going to native plants than to non-native plants. And yeah, those are the results shown here. This is also a significant result with a p-value of 0.04. But then, when we look at diversity, we can see this is not a significant result. And so this is where what I was talking about with the honeybees comes into account, where a great number of the bees that I observed were honeybees. And that's just because each colony has so many bees, and they have so many resources and are able to pollinate so much. And so the sites here, you can still see that the high peaks tend to be native plants, but they're just not quite, as significant as they were if you were just looking at the abundance counts. Something else I measured over the course of the summer was how the weather affects the bees. So I wanted to, because I know that bees don't go out in the rain, and I wanted to look at some other factors that might affect them. So sun and shade conditions over here. So this side is where there's the abundance, the richness and diversity of bees in shady conditions, and here is in sunny conditions. And so you can see it's very significantly that bees prefer the sun to the shade. So if you're planting any bee-friendly flowers, make sure to plant them in a sunny site. The wind did not have a significant impact on the bees. You can see a slight downward trend where the windier days had less bees, but it wasn't very much, so it could have just been due to random chance. And then temperature also didn't seem to have a significant effect on the bee abundance, richness, or diversity. You can see at the lower temperatures here that at 15 degrees Celsius, the bees weren't out very much, but between 20 and 30, it was pretty much just random. So they can tolerate kind of a wide range of temperatures, it looks like. So in conclusion, bee abundance and richness were significantly higher for native plants than non-native plants. So non-native plants had higher amounts of visitation and higher different amounts of species visiting. And then sun and shade conditions also had a significant impact on the abundance, richness, and diversity of the bees. Oh, and this picture I just thought was really cool, but I took it here at the hike, and it's a spider, this crab spider here eating a bee. I thought that was pretty cool.
Wow.
Yeah. This is a dog wing. Is it? No,
Wow.
but yeah, I thought that was cool. Thank you.
So any questions? What? Oh. Any questions? What was the most popular native plant though for the,
Native plant,
most popular?
for the amount of bees
Yeah.
visiting. Well, the Joe Pye weed was very popular, the dog bane was very popular, and the milkweed was very popular.
Oh, okay. So a lot of native.
Were all the bees native?
Yeah, besides the honeybee, yeah.
Okay, and they had been here a long time.
Yeah.
Did you kill the bees?
I tried my best not to.
Well,
So they were captured and they were let go. Some of them didn't work. Yeah. But I tried my very best.
what I meant to say was, what was your collection method?
Yeah. So basically, I can go back and show you the little tubes that I kept them in. Yeah. So here's a little week. So basically, I don't know what this is, but basically I would swipe, and then I have this little tube, and then it's really hard to demonstrate without a net, but then I'd try and get the bee into a small section of the net and stick the tube in, and the bee would fall in. Bees want to go up all the time, so it's kind of easy to manipulate their behavior to go where you want them to go.
Okay, got it.
Yeah.
You said there was a significant non-native plant, was that birdfoot trefoil?
Yeah.
Okay.
So that was only one site, so, wrong direction. So here, this bright yellow is the bird's foot trefoil. So you can see this one line is very high. That's on the lawn right here. So I think this is due to a lot of honeybee visitation, and I do think it's because the sun here is not blocked by any trees or anything, and they liked it a lot.
Angela, did you draw all the little illustrations?
Yeah, I drew those.
Really?
Yes.
Did you go to the collection that's here at the Huyck Preserve of the bees that have been collected by other researchers
Like,
and compare your bees with those?
yeah, so I've seen the collections. There are, so the Hymenoptera collection is all in one big box, and it's mostly wasps in there because of the work that Bob Matthews did. We heard about it earlier this summer. So there were not that many bees, but the ones I did find were ones that I also saw again this summer, so it's nice. Mhm. All right, so up next from the University of Richmond is Eli Beach Brown on his presentation on salamanders.
Hello, like Angela said, I'm Eldridge Brown. I go to the University of Richmond. Who am I? I was born and raised in Duluth, Minnesota, living kind of alongside Lake Superior. I'm currently a rising junior at the University of Richmond, studying geography and environmental science. And some of my interests include backpacking. Here's me and Daniel in the Catskills a couple weeks ago, and there was no water, but it was okay. I like swimming. There's me at Rockaway Beach in New York City. I like going to some concerts with my friends, so here's some of my friends at home. I like mountain biking, and biking, but I almost bike right into a river there. And then being from Minnesota, I really like to ski. For this summer, my work was focused on salamanders here at the preserve. The preserve here, especially this summer after the drought, had a lot of kind of much smaller streams, and salamanders generally are sometimes a primary predator in these small streams, especially streams that aren't large enough to have any fish like these streams are. As the primary predator, they have a lot to do with regulating insect populations, so a lot of invertebrates are born or spend some time in the water, and salamanders are omnivorous and they eat these insects. In doing this, they regulate the composition and basically nutrient cycling of dead matter or like detritus. And so they're key at regulating ecosystems. This is a lot of what Dr. Wyman was studying. The previous director here at the preserve did a ton of work on salamanders. And even some of what I was reading to learn about this, it was kind of cool to see him cited when I could go upstairs and read his stuff. Salamanders also are a key food source for larger predators. They live both in the terrestrial ecosystem and stream ecosystems, so they're a key food source for a lot of predators. And like I said, they live both in the terrestrial and stream ecosystem, and by doing this, they connect the nutrient webs of those two systems, bringing them together to help them kind of flow smoothly. One thing I didn't add is a lot of salamanders live a lot of their life in the soil, so by burrowing in, they have pretty large effect on soil composition. There's 18 species of salamanders in New York State, but I'll only be focusing on the 5 I saw here at the preserve. A good amount of these species that are in the state are pretty rare, and so I was unable to find them. The 5 I did see, the first 2 were terrestrial salamanders. The first is the redback salamander. This is one of the most common animals in New York State and the whole Northeast. And I'm sure if you walk around the woods enough, you've likely seen one. The eastern newt here is not fully a terrestrial salamander, but it kind of has an interesting life history. So from when it's a juvenile, it will live in the water, but then from the ages of 2 to 7, it gets this really fluorescent orange color and lives on land. So for the purpose of this study, it was considered a terrestrial salamander. My stream salamanders were the Allegheny Mountain dusky salamander, which is pretty dark with some brown spotting along the back. The northern two-line salamander, which is this really distinctive yellow color with 2 black lines, and the northern dusky salamander, which is just much darker overall, almost black all the way through. So my main research question for the summer was, how is salamander abundance, richness, and diversity affected by moisture gradients? So for my moisture gradients, I was looking at 3 different zones. The terrestrial zone, basically in hardwood forest over here on the preserve. The riparian zone, which is the zone in between streams and forests, basically the edges of streams where you see like ferns and other smaller shrubs, but not really developed forest. And then stream ecosystems. So I had 4 streams here at the preserve. I have a map that I'll show you later. And then in each of those streams, I had 3 reaches of study. At each reach, I kind of had to set up something like this with 3 30-meter transects. There would be 1 transect in the stream ecosystem, 1 transect in the riparian zone, and 1 transect in the terrestrial zone, as parallel as I could make them given the conditions. In addition to that, I had these wood blocks, basically, that look just like this. Maybe this is one. And there's 3 per transect. So salamanders like to hide under rocks, logs, and other debris. And so I used these wood blocks as a way of making sure there was an even amount or a more even amount of cover present at every site. They were pretty successful in the terrestrial zones where there was less rocks and sometimes less just cover overall. But as drought conditions increased, they became less successful over the summer. So basically what I would do is play salamander jackpot and walk the transects and flip rocks and logs until I found something or reach the end of the line. And when I find salamander, I record the species. Unlike Angela, I didn't have to bring things back to lab because there's only 5 species I showed you guys, and they're all pretty distinct. So I would record the species, the life stage, and then if I found it under a rock or a log. And then in addition to this, I would take characteristics of the area in order to look for correlations between where I found certain species or just overall abundance. And some of these characteristics included percent canopy cover, percent shrub cover, or things like leaf and moss cover. Here is a map of my sites at the hike. So we are about right here, I think. 3 of my streams were part of the 10-mile creek system. So one up here is above Lincoln Pond. The second one is right here. So basically right below the bridge. It's over there. Right below the bridge over here. And then one at the far end past the dam, like my service. And then the 4th site was about right over here as a part of Trout Creek, Trout Stream, which is a tributary of 10-mile creek. Results. My first slide here looks at total abundance by each reach, with the brown being the terrestrial, the green being the riparian, and the blue being the stream ecosystems. So to do this, I ran a statistical test called a 2-way ANOVA, and this showed that the streams had significantly more total salamanders, the number of salamanders along the y-axis. It had significantly more salamanders in total than either of the other 2 habitats. I kind of think this was affected by the drought because salamanders breathe cutaneously, which means they breathe through their skin. And as amphibians, they need their skin to be moist in order to make this happen. And so they require moisture around them at most times. That's why you traditionally find them by flipping over rocks and logs, even in the forest. But due to the lack of rain in the summer, even the rocks and logs that were pretty large and would be pretty good at keeping moisture were unable to do so just due to lack of moisture. Our Zoom video's not on here. Let's see if that'll do. Okay, whatever. Ooh. Okay. So this, like Angel said, is richness. Richness is just the total amount of species present at each reach. And as you can see, once again, the streams had significantly more species overall present. It looks as if the levels are pretty similar, but due to the fact that a lot of these reaches in the terrestrial and riparian zone just didn't have any salamanders at all, that meant the overall richness for the streams was consistently higher. And this was a significant result. An interesting change between the last two results was that diversity by reach, which is basically, as Angela said, richness taking into account the evenness of the distribution of species, was not different between the three habitats. So even though there was more overall generally more species in the stream ecosystems and more overall salamanders in stream ecosystems, these zones were dominated mostly by two species of salamanders, the two-line salamander, the yellow one, and the Allegheny mountain salamander, and due to this, their diversity was pretty similar. This is one of my only significant correlation out of all the abiotic factors I took regarding my site, and this was with shrub cover. I found that shrub cover was highly negatively correlated with the total amount of salamanders. So on this graph, on the y-axis here, this is the number of occurrences of that amount of abundance. So it's kind of confusing, but basically how many times this amount of salamanders is found at a reach. In the x-axis was the amount of shrub cover. So as you can see, there's pretty strong negative correlation between a lot of shrub cover and a high amount of salamanders. I'm not exactly sure why this is, but I think it might have to do with just like available food or the amount of like leaf litter covering the ground, or just cover overall, but I'm not exactly sure. And this analysis was done using experience correlation analysis. These are two more correlations. While these correlations were not significant, they still kind of help explain the data. This correlation was between the abundance of salamanders and downwoody debris, with the meters squared per hectare, or just basically the amount of downwoody debris on the y-axis, and the x-axis being the amount of abundance. Downwoody debris is basically any dead, fallen logs or trees that have kind of decomposed a little bit. And I had actually predicted this would be the opposite. I had kind of hypothesized that the more downwoody debris there was, the more salamanders you would find, because it's good habitat for them, especially in the forest where there's not that much. But there was kind of a pretty moderate negative correlation. One other positive correlation was between leaf cover and abundance. So basically this was a 1 through 5 scale of the amount of leaf cover on the forest floor. And I found that leaf cover was positively correlated with the amount of salamanders. That to me makes a lot of sense, because having leaf litter in an area brings with it like insects that like to live in that leaf litter, and this provides good food, and the leaf litter provides food as well as helps to trap moisture, which the salamanders need. Conclusions. There are generally more species and more total salamanders in streams due to need for moisture. Like I said, I think this summer was possibly related to drought, so it would be something to consider again. And like I said, the stream sites were highly dominated by the two species, and as such, the diversity was equal over the gradient. And factors such as shrub cover and the amount of downwoody debris negatively impacted the amount of salamanders at a location, whereas factors such as leaf and log cover positively affect the amount of salamanders found at the location. And that is all. Any questions? Yeah.
I'm just wondering, since they're partly or a good deal terrestrial, what accounts for that bright orange color, making them, easier?
Bright orange color in those specific salamanders is a measure of their toxicity. So it's basically a mechanism for them to signal to other terrestrial creatures like, don't eat me, I'm poisonous.
Oh.
It's like if I was wearing a pink shirt. Yeah.
Did you see any sign of bear activity in the areas that you were surveying? In other words, let me suggest that one of the things you may have seen without recognizing it is turned over logs, freshly turned over logs.
I would say no. I didn't see any like bear scat or any like scratch marks on trees or anything like that. That looked like a larger creature had done it. I don't know why. We saw a bear outside the bathroom, but that was it. Yeah.
Other than shifting towards the stream for these terrestrial salamanders, are there other refugia available within the forest landscape for them to hunker down in these drought conditions?
Yeah, my guess is a lot of the terrestrials. So basically, I didn't really see a movement of the terrestrial salamanders towards more high moisture areas. I basically just saw less of the terrestrial. If I had to guess, I would say most of the salamanders here do burrow for some portion of their life, some more than others, and I would guess the terrestrial salamanders then took to burrowing more as a way to get to higher moisture areas instead of moving into another area that is already occupied by other salamanders. But I don't think like the terrestrial salamanders would be able to outcompete the stream salamanders in the stream, so they wouldn't have been able to function there well. Yeah.
That actually leads to my question. I believe on field trips we have seen salamander burrows under logs. Did you notice, were there such things, and maybe the ones that you thought might be under the log were actually just further into the soil under the logs?
Yeah, I would say I didn't necessarily see the burrows sometimes, but especially with the redback salamanders, which is the main terrestrial species, I might flip a log and it would be there and it would just go into the ground. I don't think the entrances to their burrows are probably large enough for me to notice commonly, but I think they definitely did exist because sometimes they would just be gone. I'd go like right to the down and it's just disappeared.
Yeah.
Cool beans.
Okay.
Next up is V. She's studying macroinvertebrates in the stream, and she is from Soka University.
Okay, thanks Eli. So I'm Tanvi, and I'm going to present on decomposition and macroinvertebrate presence here at the Huyck Preserve, which comes under freshwater ecology. So a little about me. I come from New Delhi, India, south of New Delhi, and I'm studying biology in Soka University of America, which is also in really Southern California. And I like being outdoors, and I like walking outdoors, being outdoors, and I love all sorts of animals. This is my dog. He's a dog cat. Okay. So before we jump in, I want to talk a little about the aquatic macroinvertebrates that I study. So what are they? They can be insects or not, but they're animals that spend at least some portion of their lives in water. So a lot of flies, like crane flies, dragonflies, their larvae spend their lives in water and then they mature and fly out. They are visible to the naked eye and they lack a spine. And these are some of the common invertebrate families I found here at Huyck. So this is a stonefly naiad. A naiad is basically a nymph or a larvae. And this is a dragonfly nymph or naiad. This is a scavenging beetle. This is the crayfish, a big water crayfish. That's the most common crayfish we have here. It's not invasive. And this is a helcromite, also known as a dachshundfly larvae. These can get pretty big. So they actually play a really important role in the freshwater ecosystem. So the most important role they play is nutrient recycling. So they eat a lot of detritus and they're decomposers, and then they excrete back the leaf litter that they eat. They release nutrients like phosphorus, nitrogen, carbon back into the ecosystem. And these nutrients are limiting nutrients. That means plants and animals need them a lot for growth, but they're present in very limited quantities. And they're really hard to absorb. So macroinvertebrates release them back into the ecosystem. And this way, they directly impact food webs, and primary production of plants and algae. And then they're a really important part of food webs because they're predators, smaller invertebrates, and they're prey for higher trophic level organisms, from birds to birds and amphibia and even other macro invertebrates like crayfish. And thirdly, there are indicators of water quality. They are commonly used for assessing stream quality. And this is a really cool poster that shows that the common macro invertebrates we know can be divided into 3 groups. And group 1 invertebrates are extremely pollution intolerant, so their presence in streams is generally a good indicator that the water is of good quality. And these third group are indicators of poor water quality, and the middle group is somewhere between the two. So if you see like leeches and midges and mosquito worms in the stream, then it's an indicator that the water is not doing the best. So I had 3 research questions, and so my first research question was, is there a significant difference in the amount of decomposition going on in leaf litter between 3 differently sized streams, and mesh size and number of days? I'll come to that in a bit. This is my most important question, my main question. The second question was, is there a significant difference in the number, richness, and diversity of aquatic macro invertebrates that I found in the 3 different streams? And the third one was, is there a correlation between the number of invertebrates I'm getting and the decomposition of leaf litter that's going on? And these are my sites. So this is my site number 1, which is, I shared 3 sites with Eli, and this first one and third one are located on the 10-mile creek, and the second one is part of the Trout Creek, I believe. So, and this is like on the south of the Master Dam. And these 2 are just like right here. The first site, first stream, is really narrow and it's really shallow and it's really slow. And the second one is medium and all these things. And the third one is deep, it's wide, and it's really fast flowing. So I had all these 3 streams, and each of these streams had 3 reaches, and each reach was 30 meters long. A reach is just a segment of the stream that I'm studying. And each reach had 4 sites, so a total of 36 sites. So the first thing I did was look at stream characteristics, and some of the things I looked at were the size of the stream, the flow of the stream, the depth of the stream, canopy cover. Here I'm looking at the canopy cover using a device that's called spherical densiometer. Looked at what kind of plant species are around in the overstory, understory, and the substrate of the lake. Here I'm measuring the width, and here I'm measuring the depth. Okay, to study the decomposition rate, I took mainly 2 sizes of mesh bags. One is 1 millimeter big, and the other this is 1 millimeter mesh bag, and this is a 7 millimeter. So the reason I did that is because certain larger invertebrates cannot get into the 1 millimeter bag, so I could rule out that the leaf litter loss that's happening in the 1 millimeter bag is not because of the bigger invertebrates. And I filled them up with 5 grams of dried leaves from the dominant tree species around here, and I suspended them in water like this with like rocks and flags all over my sites, and I kept them in first for a period of 7 days and then a period of 14 days. Those periods of time did not overlap. And at the end of the time, I took them out and I opened up the bags, took out the leaves, dried them, weighed them, and also collected any invertebrates that were in the bag. For the second part of my experiment, I did kick sampling. This was because the water level was okay, and I was able to put the net like kind of facing like downstream, and then I could kick the water, and all the invertebrates that were stuck to rocks flowed into the net. And then I poured it out on a tray, sorted and threw it, looked for any invertebrates, collected them in glass jars, brought them back, and identified them to their family. But by the end of my project, the water levels were really low, and I had to do hand sampling, basically move the rocks by hand and do that. So my results. So I did a lot of statistical tests, both for significance and correlation. So I'm only going to show the ones that I found significant data for. So here the first one is the 3-unit ANOVA, which is a test for significance. And I have my stream size, so the x-axis, and the y-axis is the average leaf loss I've found. And the colors are 7 days and 14 days, as you can see. And over there was the leaf loss between the three streams was highly significant for a value less than 0.01, and these two are not that significant, but small and medium are both significantly different from the big stream. What's interesting to note here is that the big stream, which was the deepest and had the fastest flow, saw the least amount of leaf loss. And I think this is because the water was flowing so fast that all the macroinvertebrates were kind of swept away. So they didn't really get that much time to decompose the leaves. And something interesting is that if you see the leaf loss basically almost stops after the 7-day mark. And in the big stream, it's actually less for 14 days than 7 days. So that's also really interesting, which tells us that leaf loss really almost stops after the first 7 days, when decomposition is the fastest. And this, this graph is also correlates with the other graph I show you. This is about decomposition, that is the leaf loss. So this is average daily decomposition, and 7 days has much higher decomposition rate than 14 days, just because the amount of leaf loss was almost the same for both number of days, but the amount of time period was longer for 14 days, of course. And yeah, so these were my main ANOVAs, and I also did a post hoc test, which is to kind of find out which streams were actually different. So small was highly different from the big, and the medium was highly different from the big. But the small and medium were not that highly different because they're more similar to each other. And I did a bunch of correlational tests, but these were the ones that were significant. With increased flow speed, there was a decrease in leaf loss, again, because the macroinvertebrates were being swept away. But with the increased average flow speed, I found increase in richness as well as abundance. And I used Pearson's correlational analysis test to make these scatter plots. And finally, the stream quality. So leeches, leech and midges are indicators of poor water quality, and I found a lot of them in the big stream, which is at the south of the dam. But the first two streams, small and medium, lacked leeches and midges, and had tons of water pennies and helper mites or Dobson-fly larvae, which are, quite pollution intolerant. So it means those streams are doing really well. So in conclusion, leaf litter decomposition was highest in the smallest, shallowest, and the slowest stream because it had just in general more invertebrates than they were able to collect together. Also, the leaf litter decomposition slows down the time. The first week sees the highest decomposition, and this is corroborated by previous studies. And my second and third research questions are really long, which was I didn't find any correlation between average decomposition and macroinvertebrate presence, but this doesn't mean that there is no correlation. I just didn't find it in this study at this time. And similarly, I also did not find any difference between the three streams for macroinvertebrate abundance, diversity, and richness. So that's it. Thank you.
You talked about how the biggest stream was the least healthy due to what you found there. You also said the biggest stream had the lowest amount of decomposition. Do you consider, or do you think it's possible that the pollution intolerant species maybe have to invest more resources in being able to pollute or tolerate that pollution and therefore are unable to decompose as fast?
No.
No.
No. Thanks for asking. No, it's a good question. I found a ton of invertebrates there, but they were quite different from the ones I found in the first and second stream. But still, there were a lot of different kinds. The problem wasn't that the diversity and abundance or the type of the macroinvertebrate. In general, it was just, I think, was the speed. But again, I'm just saying this off the top of my head. I will have to study further and see what was the actual reason.
Okay.
Yeah.
So your first two sites, the two smaller and medium sites, are above Lake Minnesotis, and the large one is below.
Yeah.
Do you think that the difference in water quality as indicated by the inverts might have something to do with the impoundment?
It could be, for sure. So all the three streams are part of the same creek, the Townline Creek. No, I think one of them is from the trout, but yes, definitely could be that. It's also like a really shaded area. And yeah, it's not very, yeah, it's like these two streams are very well placed and they get like good amount of sunlight, and I think they're just much also cleaner. Like that one down there is kind of unkept at this point, like, yeah, so I think that's definitely something to do with that. Also, I think the temperatures might be different because that one is really shaded.
Go ahead.
I don't know if you go to me or my husband here. He's the big mouth. Okay, so was there a greater abundance of pollution tolerant species as the drought progressed? Did you find that increased as it got drier?
No, it was, no. So as it got drier, I had to resort to hand sampling, but the abundance of species did not change with time. Yeah, I got like equal number of species both times because as the water level went down, the animals kind of congregated in areas that were at that deeper water level. So the abundance really did not change with droughts. That's a good question. Thank you.
Okay. I get a chance on? This is a question that I should have been thinking about asking all of the previous speakers as well, and that is, did you do a literature search
Yes.
before you undertook this study, and particularly pay attention to the work that's been done here on the Huyck Preserve with respect to the topic or similar topic that you're studying?
So I did do literature review before, and so most of my methods I picked out from the literature review. And so I did not know that research of this sort had happened at Huyck before. It's really funny because I was looking for material to make my bags, and Adam, the groundskeeper, he told me that someone did a similar kind of study 25 years ago. But that's when I found out about it, and I was like really deep into my research at that point. So, but yeah, it would be really interesting to look at what kind of data they got.
Okay, thank you.
Next up is Daniel Graves from Brown University, who's half of white. Soil.
Slow like drip or not when they are urban mustard.
Okay, awesome. So my name is Daniel Graves, and I studied soil and earthworm change over successional gradients this summer. And so yeah, before I deep dive into that, a little bit about me. I am from Vermont. I live right around here, southwestern part. I really like snow. I go to Brown University. I'm a sophomore, graduate junior, and I study environmental science there. So. Okay, like Eli, I'm also really into hiking, and That's me in the Green Mountains of Vermont. I'm also really into puzzles, speed puzzling especially. This is me really fascinated with the amazing game of baseball. It's me fascinated with the snail. And this is me looking really cool. So yeah, earthworms. So this summer I studied earthworms, and on the Huyck Preserve, pretty much all of the earthworms are non-native and actually cross most of North America. Earthworms are mostly non-native. There are a couple that are native, but they mostly stay to like swamps or and wetland areas, but I didn't study those areas, so I didn't find any of them. But there are also some invasive earthworms, such as like jumping worms. I also didn't find any of those, which is good, but in general, earthworms, because they're not native, can be really impactful to the ecosystems in the forest here. So first of all, they speed up decomposition because they're eating the leaf litter and converting it into nutrients and such. They can compact soil, increase erosion, and where they're native, that's not really an issue because the soils are adapted to that and the forests are adapted to that. However, here, a lot of the tree species aren't capable of taking up those nutrients as quickly, and so that might get leached out of the soil. On the Huyck Preserve, I mainly saw two genera of earthworms, Aphractodia and Lumbricus. And so these are Aphractodia earthworms. They're typically pretty small and they don't have any color pigmentation. It looks like they're pink, but that's really just their internal color. They're maybe from 1 centimeter to 5 centimeters typically. And then these are Lumbricus earthworms. They are pretty common. They're what we might know as night crawlers, often used for fish bait. And they can be up to 10, 15 centimeters long, and they're much darker in color. And yeah, so I can kind of identify these sometimes without a microscope, but usually I do have to use a microscope to identify the species. So this summer I studied how soil characteristics and earthworms change over successional gradients. And by successional gradient, I'm basically looked at 3 different stages. For fields, young forests, so I call those successional forests, lots of ash and maple, and then mature forests. And I only looked at hardwood forests because having especially conifers changes the soil characteristics, especially pH, because they're much more acidic. So for my hypotheses, I predicted that earthworm density would be higher in soils with more moisture and cooler temperatures. Just thinking earthworms probably moist environments. I predicted soil with more earthworms will have higher nutrient levels because they're kind of cycling those nutrients in from the detritus. Earthworms will be most abundant in older successional stages, and soil in mature forests would be colder and have more moisture than younger successional stages. And I think this one is fairly logical because if you have more canopy cover, there's less sunlight reaching the bottom of the forest floor, and so it would have more moisture and colder temperatures. So what I did to sample this was took 3 transects with fields, young, successional forests, and mature forests. And so this is a map of the preserve with all the different ecological communities. And the yellow is the fields, so these 2 fields. There's another one up here that's blocked, but it's way up the northern end of the preserve. So the fields are the yellow, the light green is the successional forest, and the darker green is mature forests. And these are slightly different dominant tree cover in these 2 mature forests, but they have pretty similar compositions. So in each of those sites, I would have 5 different sample points. So in total, there's 15 per transect, and then 3 transects, so 45 sites. And at each site, I would test for the soil, different soil characteristics. So first off, when I was in the field, I would look at the temperature and the different soil horizons. So really here there's not much evidence of soil horizons, and that's likely because they're pretty young soils and they often have been plowed within like maybe the past like 100 or 200 years. And so plowing like mixes up the soil and makes it so that those horizons that build up over hundreds and thousands of years kind of get mixed up together and aren't there anymore. So I'd look at horizons, but I didn't really see any. And then I'd also take a soil sample back to the lab where I would test for pH, moisture, nitrate, phosphorus, and potassium. And moisture I just took the wet weight, dried the sample, and then took the dry weight and found the difference in mass. pH, nitrate, phosphorus, and potassium were all test kits that I used to just test the different chemical properties. And this is the test kit used for the nutrient sampling. And you kind of just put the sample in with a little of the test solution, and it will turn a different color based on how much is in the soil. And also at each site, I would sample for earthworms using the mustard method. So there are two main methods of sampling for earthworms. The first one is using mustard powder. So I take 20 grams of mustard powder and 2 liters of water, mix it up together, and use my bottomless bucket to delineate the area, pour it in and see what comes up. Usually they'll pop up pretty quickly if they're there, as long as the water is saturating the soil. And then I'll take those back to the lab. The hand sampling method is also pretty common. It's just really just digging into the soil. And I did that a couple times just to make sure I was getting the same species, but I didn't do it enough to have any results. And then in the lab, I would try to identify the earthworms, which is what I'm doing there. So it's kind of fun to see what these earthworms look like when they're covered in the mustard powder. The mustard irritates their skin a little bit, so that's why they want to come up, and they'll wriggle around a bit. So, that's sideways, but that's an earthworm right there. There's another one somewhere here, down here. But yeah, that didn't work very well. Let's see if this one's better. Yeah, so down right here, there should be one large earthworm coming up. Yeah, and they can kind of really shoot out of the soil, which is fun to watch. Yeah. So, yeah. Okay, that's a little off-center, but I think it has everything I need.
Show me
I think I skipped a slide, but so this is, I'll just go over this. This is soil moisture by successional stage, and it's pretty significant results here, especially for the field. So this is the percent moisture in the soil. In the field, it was a significantly lower result, and it kind of just decreased with basically the amount of or how young the site was. But yes, the only significant result here was fields. And I think I skipped pH, I'm not sure why it's not going back, but pH was similar in that the mature forests had a lower pH, which was a significant result. But the successional and fields were pretty similar. Although one interesting thing was that the spread in the fields was much larger. And I think that might have something to do with because since they're younger sites, they might have more impact from their direct history of land use. So some of them might have been plowed recently, or some of them might have been pasture. So I think that could have had an impact in how the different fields have different pH levels. Oh, no, I was messing with that. So yeah, pH very different in the mature forests than in the fields and successional. And yeah, so this kind of just shows that the spread is much larger in the field than in the other two sites, and it was significantly lower in the mature forest. I also looked at temperature by successional stage, and in the field and successional forest, they were pretty similar. It's interesting because in the successional forest, a lot of them are dominated by ash, and since the emerald ash borer is like impacting a lot of the ash populations here, there is pretty low like canopy cover there, so that's probably influencing how warm the soil gets. Whereas if it was just a normal canopy cover in like a sapling forest, it probably would be a little colder. Not sure why it's doing that, but this is looking at the nutrients. So this is nitrate and potassium by successional stage. Blue is potassium, yellow is nitrate. And as you can see, it's pretty much like there's almost, at the lowest level, there's almost no evidence of these nutrients. And I even tested them with 2 different test kits, so it's not like an error there. And so that just probably indicates that these soils have very low nutrients. And it's similar with phosphorus. It's really hard to see, but there's a little bit more variation there, but still very low, and no significant difference. So as far as the earthworms that I found, like I mentioned, there's 2 significant genera. The green is Lumbricus, the blue is Aprotodia. I found 2 earthworms of a different genus, Icenia and Dendrobaena. So I found 1 instance in mature and field for Icenia and Dendrobaena, respectively. There's pretty much no difference between Lumbricus across the 3 gradients. However, Aprotodia was found much more commonly in mature forests. Although that wasn't a significant result, it's pretty clear that there's quite a few more in the mature forests. And then the last thing I kind of looked at was the correlations between earthworm density and some soil properties. So firstly, with soil moisture, there's not too much of a significant, or there's no significant correlation, but a slight positive correlation between moisture. With more moisture, there's slightly more earthworms, both in Lumbricus and Aprotodia. And then with pH, there is a significant difference as far as the Lumbricus. And this is, I followed up with some literature review on this, and Lumbricus really don't like acidic soils, and that is exactly what I saw. When the pH was low, I found almost none of them, and that was a very significant result, but that didn't seem to affect the Aprotodia at all. And so in summary, mature forests had soil pH and temperature significantly lower than in the other 2 stages, and soil moisture increased with the age of each stage and was significantly lowest in the fields. Aprotodia density was higher in mature forests, but that wasn't significant, and Lumbricus density was significantly correlated with pH, preferring alkaline soils. And then, like I mentioned, there were a number of other soil characteristics I looked at, but they were not really significantly correlated with earthworm density. And I'm not actually throwing up into this bucket, I'm just looking at it. Yeah. And yeah, that's us. So, thank you.
Question.
You said there were no native New York State worms. Okay.
Yes.
So are these all ones that came over from Asia on boats or what?
So most of the ones I found are from Europe. So Lumbricus and Aprotodia are both from Europe. The jumping worms are from Asia, and I didn't find any here on the preserve, but they are in the region, and they're pretty. You can tell pretty easily the difference between the jumping worms. They have like the hair pattern is quite different, and they move around a lot more. They're much more active. But yeah, I don't find.
But they're all from Europe?
The jumping worms are from Asia. The rest of them are pretty much all from Europe. Yes.
Are you familiar with the technique of collecting worms by introducing electricity into the ground?
I have not heard that, no. Have you,
Well-
have you tried it?
I can tell you that it works because when we were kids, they were turning out these little, the Army had little generators that you could hand crank. And you'd have two probes you put in the ground. And you crank that and your worms would come up.
That's wild.
Think about using
I
that.
wanna try that, just for fun.
Of course, in the drought, with no water. Yeah.
Well, you could introduce the water, I suppose, right? He was dumping water in his bucket.
Yeah, one thing I should note about the drought is that I sampled the earthworms a little bit earlier in the summer when there was a little bit more moisture. This week with the high schoolers, I sampled some earthworms with them, and the soil was so dry that, and Sue has talked about this, like with air bubbles in the soil, it's hard for water to kind of penetrate into the soil. So we were basically only getting an inch deep into the soil with our samples. So this week with the high schoolers, we switched to doing hand sampling, and that seemed to work better. But when I was sampling, that wasn't an issue. Yeah.
One more, what is the major predator on worms that are in the Huyck Preserve? Do you have any idea what that is?
That's a great question. I'm not really sure. All I know is that I see birds sometimes eating them in yards like llamas, but
I've wondered about
Yeah.
ants.
ants.
Yeah, ants, because, you know, they're pretty aggressive.
Yeah.
And they're in the same area, you know, proximate to one another.
Yeah.
And, so, when you put them together, what happens, you know, I mean, it's the question.
That's a good question. I can tell you that we sampled one spot with an ant colony right next to us, and we still found earthworms. But that's all I know. So, yeah.
Can I do a weird question? Okay, and you may not have studied it or not, did you notice like an increased correlation with skeletons or more decomposed leaf litter in the areas where there were more earthworms?
It was like I did look at leaf litter, and there was no like significant correlation. That is the topic that the high schoolers chose to look at. I haven't talked to them about the results, but I don't think they found any significant correlation. I think when you have jumping worms, that is because they're so much more active. They will decompose the leaf litter much more, and so it's really noticeable. But I don't think with the worms we have currently here.
Okay.